So my name is Mattias Ulen. I'm a professor in biotechnology at the Royal Institute of Technology. I'm also the founder of the Human Protein Atlas, which is sort of headed here from Stockholm. And also I'm the chairman of the board of Atlas Antibodies. So there has been a lot of editorials, but also articles written by people in the research community questioning some of the scientific results and the reproducibility of scientific results published even in very prominent journals. And of course, antibodies is key for this, that we need to trust the antibodies we have, and we need to trust the validation that the antibodies has gone through. And it is very important then that we should make sure that the reagents that are used for scientific research is actually working as intended. And what we have focused on then is the validation of antibodies, to try to make sure that when we're using an antibody, it can be used for a specific application and a specific context, and then actually bind to the target that is intended. It is very critical that we can trust the antibodies used in research. If they are not of high quality, we are wasting time, we are wasting resources, and we are wasting money for the users and the funders. So it's absolutely essential that we can actually trust the antibodies, and that's why it's so important that we have guidelines for how to validate antibodies. So we decided to form an international working group for antibody validation, and this group of scientists worked for a little bit more than a year to try to then formulate guidelines, and this was then published in a perspective article in Nature Methods. So what is the depth that you need to validate an antibody? And what we have sort of come up with is minimal requirement, show all the primary data, but then on top of that try to do validation according to IVGAV. We call it enhanced validated antibodies. So in the IVGAV, we actually then described 5 pillars for validation. The first one was to use genetic methods, that is to actually decrease the target protein by genetic means, such as CRISPR or siRNA. The second one we now call recombinant expression, that is that you actually increase the level of the target protein, and this way you see that the staining then is increased. Another interesting way of doing it is to use independent antibodies, and that is to have at least 2 antibodies binding to the target. And this way you can compare the results of these 2 antibodies and then validate one antibody with another antibody. And then you have the orthogonal validation, which is actually using an independent methods, which are not using antibodies, and then compare the result from that method with the antibody-based method. And finally, you can also use mass spectrometry, capture the target, and in different ways then analyze the results. So altogether we have 5 pillars: genetic, recombinant expression, orthogonal validation, independent antibodies, and then capture MS. We're now using and adhering to the IVGAV rules for validation. And when they have been validated using these methods, we call them enhanced validated